Hello, my dear students. Welcome back to our offline classes. It is quite exhilarating to be back among you, away from the scare of Corona and devoting some productive time to our lecture series. To begin with, the first lecture is concerned with chromosome centromere and fidelity of segregation, that is, how precisely the chromosomes are segregated during the cell division. So let us begin with the screen sharing. So centromere and segregation fidelity, part 1. The cells continue to divide, although many cells stop dividing too, but most of them continue to dividing to make up for the growth and development of our body, as well as to replace the cells which were destroyed during the wear and tear of our activities. So what is particular about the cell division? That is, the cells must give rise to daughter cells having similarity, particularly genetic similarity with the mother cells. That is, the offspring should have same number of chromosomes. The daughter cells should have same number of chromosomes which the parent cells were having, and the chromosomal faithfulness, preciseness, and in other words, fidelity should be maintained among the daughter cells too. If a chromosome is destroyed, a part of chromosome is destroyed during segregation, what will happen? Aneuploidy will result, and that cell will lose many of its functions, and it will behave as a raw cell, as an abnormal cell, as a cell out of control. So what is most important is that the cells should maintain fidelity. That is, the division should be very precise. It should show exactness. Exact number of chromosomes should be passed to the offsprings, and that is the segregation facility. You see here the majesty of cell division. The cell is dividing into two daughter cells. You see here that the chromosomes are arranged along the equator of the cell, and here it is the centromere. You see here it is the centromere, and these are the spindle fibers converging upon the two spindle poles, centrosomes, you say. So these spindle fibers, they pull the cell, pull the chromosomes which have been arranged along the equator towards opposite poles. Say, for example, if this particular attachment did not materialize, what will happen? This chromosome will not be pulled apart to this pole. So this dividing cell will have one additional chromosome, and it will create all sorts of problems. It will create an aneuploid cell, and that aneuploidy will change the behavior of cell, and the cell will go haywire. So what is important, what is of prime importance, or what is significance of chromosomes is that it maintains the chromosomal fidelity. It segregates the chromosomes in a fiddle way, in a faithful way, in an exact way towards the two poles, so that the two dividing cells, that is the daughter cells, will have the same number of chromosomes and they will have the same set of chromosomes, you can say. So let us begin with centromere and chromosomal segregation. As I told you that proper segregation of chromosomes during the cell division is of paramount importance, because if any discrepancy sets in, one cell will have more number of chromosomes, one of the daughter cells will have more number of chromosomes than the another daughter cells, and in effecting this segregation and in maintaining its fidelity, the centromere has got an important role to play. So what is centromere? Centromere is a primary constriction, a constriction along the chromosome which holds the two sister chromatids together. Sister chromatids results after the S phase of the cell division, and when the cell enters into the M phase, the chromosomes will have two sister chromatids. They get attached at the point where the centromere is, and so this constriction is called primary constriction, and the point will have assembly of proteins to form the centromere. So at this point of centromere, kinetochore, a disc-like protein kinetochore, will be attached, and to that kinetochore, spindle fibers will come to attach, and those spindle fibers will pull the chromosomes towards the opposite direction, opposite poles, so that the complete set is made available to each of the dividing cell. So eukaryotic centromere is a specialized protein-bound chromosomal region. It is a region on the chromosome which is called primary constrictions, where the sister chromatids get attached to each other, and it is protein-bound region. It is attached with characteristic proteins, and those proteins play further role in the attachment of the kinetochore to this region, and to that kinetochore, the spindle fibers attach, and those spindle fibers emanating from the two poles pull the sister chromatids towards the opposite poles, so as to make available a complete complement of chromosomes to each pole, which results into the presence of a complete set of chromosome in each of the daughter cells. So a specialized protein-bound chromosomal region to which kinetochore is attached, and its length varies from 110, 125 base pairs to thousands of base pairs. And so what about the maintenance of segregation fidelity is that centromere, which is a protein-bound chromosomal structure at the primary constrictions, get attached to the kinetochore, to which the spindle fiber come to attach, and this arrangement, this attachment and subsequent pulling maintains the segregation fidelity of the chromosome. So here you have a basic structure of centromere. These are the two sister chromatids. You see here, there is a constriction at this point. The two sister chromatids are attached to each other. So here you see the centromere having attached protein, and to this centromere, here disc-like protein called kinetochore, they come to impinge upon the centromere, and to this kinetochore, spindle fibers are attached, and they will pull the chromosome, the sister chromatids, into opposite direction. If there is any malfunction in attachment, the centromere kinetochore arrangement will not allow the chromosome to be pulled apart. So it is not a passive attachment point, rather it regulates the fidelity of segregation. That is, if attachment is not complete, or if there is some malarrangement in the attachment between the spindle fiber and the kinetochore, the entire process of segregation will be halted, and it will be checked and rechecked again to ensure that what has went awry, what has went gone, and then after rectifying the mistake, the process will be further proceeded. That how this process goes on, we shall see in the latter slides more precisely into the part 2. So shifting to the next slide, here we have seen in detail the structure. So here you have got the sister chromatids constricted at primary constriction, the centromere, the primary constriction along with the proteins. These constitute the centromere, and at the centromere, this kinetochore has come to attach, and you see the spindle fibers emanating from the poles. These fibers extend and attach to the kinetochore of the centromere. So here the process is beginning. You see in this process had been highlighted here. You see these spindle fibers, they have not reached, but this spindle fiber has reached the centromere, to which kinetochore is attached, and it has got bound to the kinetochore. Now you see this arrangement, the pulling process. When the binding has been perfect, the pulling process in the opposite direction has begun, and this process is the metaphase of the M phase, during which the chromosomes are bidirectionally arranged. That is, they are arranged on equator, and they are attached to the kinetochore on either side of the equator. Now you see here that one of the spindle arrangement, that is the set of fibers, they have pulled. Here you see that in this direction and this direction, one of them is being highlighted. So here it has begun pulling the chromosome towards one of the poles, and due to this pulling, the chromosome has assumed a V shape. So the chromosome, which was almost straight, has now, when the pulling has begun, it will assume a V shape, and the other side will also assume a V shape, as you see in this diagram. So coming to the details of the arrangement, you see this is the DNA. These are the nucleosomes, DNA wrapped around the nucleosomes. So this is chromatin of the centromere, that is, chromatin is DNA plus protein, histone proteins. So you see here DNA is wrapped around the histone core, the octamer, and to this centromeric proteins have come to attach. So this arrangement is centromere, and to this centromere, you see here kinetochore proteins have come to attach, and to this kinetochore protein, spindle fibers, which are made up of microtubules, they are arranged. So this arrangement had been highlighted here, or this arrangement had been highlighted here. So centromere constitutes of a segment of chromatin which has been bound to the centromere proteins. So it is a protein-bound DNA structure or protein-bound length of the chromosome to which kinetochore come to attach, and to this centromere kinetochore arrangement facilitates the attachment of the spindle fibrils to the kinetochore, which subsequently pull the chromosome towards the poles. And these sister chromatids, when they are pulled towards the pole, they will occupy a place on the either side, that is in the two of the daughter cells which are dividing from the one mother cell. Going ahead, CNPA. What is CNPA? So I have taught you in chromosome classes that the chromosome is basically made up of nucleosomes. What are nucleosomes? Nucleosomes are DNA wrapped around histone cores. What is histone core? Histone core is an octamer. This octamer is made up of histone, which are basic proteins, and it is made up of H2A, H2B, H3, and H4. So there are 4 types, and one H1 is linked to the linker DNA. So here we are concerned with the octamer. So octamer is made up of 2 molecules of H2A, H2B, H3, and H4. So this is in normal case. But here in centromere, there is a difference. That is, the centromere differs from the normal histone octamer or the normal or the canonical, which you can say normal or canonical histone core, in which H3, H4 tetramer is formed, around which H2A, H2B dimers, 2 dimers got attached. So here in normal cases, in canonical cases, in canonical chromosome, in canonical nucleosome, what happens? That H3 and H4 are involved. But here at the centromere, the H3 is replaced by another protein, another variant of the histone, which is called CENP-A, centromere protein A. So what is centromere protein A? Centromere protein A is a variant of H3, which had been variously named in case of different animals. So centromere protein A, it's called CNP-A in mammals, CID in flies, CNP1 in Saccharomyces cerevisiae, that is the fission yeast, and it is also called in budding yeast, it is called CSE4P. So there are 2 yeasts, the budding yeast, Saccharomyces cerevisiae, and there is fission yeast called Saccharomyces pombe. So in Saccharomyces cerevisiae, the CSE4P is nothing but a CNP-A, that is a variant of histone H3. The histone H3, canonical histone H3, or the usual histone H3, which is found in the nucleosome elsewhere, had been replaced by a variant which is called CENP-A, centromere protein A in mammals, but it has got other names in other animals, such as the CID in flies, CSE4P in budding yeast, and CNP1 in fission yeast. So it would be better to call CEN-H3, that is centromere H3, that is variant of H3 which is found at centromere, and it will mean all these 4 names. So you should not get confused about the names. Many of you have asked in your feedbacks that what is CID? So CID is nothing but the variant of histone H3, which is present in the Drosophila. So what marks the centromere? Centromere, as I told you, is a length of chromatin, is a portion of chromosome. So which portion of chromosome? As I told you earlier, that the chromosome, portion of chromosome is specifically attached to certain proteins. What are those certain proteins? So the first and foremost, that is the attachment of one protein which transforms the part of chromosome into centromere is centromere protein A, or its variants such as CID, CSE4, or CNP1. So when this CNP-A or CEN, centromeric histone 3, so you should always be clear about canonical histone 3, that is histone 3 found elsewhere on the chromosome, and CEN-H3, histone 3 found at the centromere. So you should be always have a clear conception of CEN-H3, that is CNP-A or CID or CSE4, that is CEN-H3, centromeric histone 3, whereas canonical histone 3 is the normal, the usual histone 3, which is found on the chromosomes, in the nucleosomes of the chromosome elsewhere, that is other than centromere. So you see here that there is H3 variant, canonical H3 elsewhere other than centromere, but at the centromere there is a variant of histone 3, H3 variants, which is called CNP-A, and its homologues dictate the location of centromere in a DNA independent fashion. So whatever be the composition, whatever be the sequence of nucleotides on that portion of the DNA, it doesn't matter. It is the attachment of the CNP to a particular sequence of the DNA that imparts the centromeric property on that particular DNA. So H3 variant dictates the centromeric location, which is DNA sequence independent manner. That is, it hardly matters what is the sequence of the DNA there. It only matters that the CNP-A or other variants analogous to it have got attached to that point. It's important in centromere specification got from silence centromere. I have got a question from you regarding silence centromere. Priya Singh, roll number 44, she had put a question that whether the silence centromere is like the recessive site, recessive locus of a chromosome. No. You should not get confused. Silence centromere, what happens that sometimes centromere shifts its location from its usual position of the chromosome. So when a centromere shifts its location, the previous location does not act as a centromere, and that previous location is called silence centromere. Although this process is not usual and it will lead to changes, genetic changes, and it is in most cases it is harmful, but it has also contributed in evolution. Particularly it has been studied in the evolution of ecosystem species, which we shall be dealing in the part 2, or it is helpful in speciation. So from the individualistic point of view, this silencing will be deleterious. That is, it will have harmful impact. But from the evolutionary point of view, that is from generational point of view, it has got some impact on speciation and evolution. So it has nothing to do with the dominant and recessive character. Rather, it is shifting of the centromere from its usual location that leads to the silencing of the previous centromere. That is, the centromere, the location which was previously acting as centromere now gets silenced. So we were dealing with this DNA. What is the important? I have said that it is the centromere marker. So whenever centromere shifts its position, although we shall delve deeply into the structure of centromere in later slides, but here I will mention in a passing manner that centromere usually have repeated DNA sequences. So DNA sequences there at the centromere are tandemly repeated. In case of man, many megabases repetition occurs. And so, and to that tandemly repeated sequence, the centromere A is attached. CENP-A or CEN-H3, centromere H3 protein gets attached. So at normal centromere, repeated sequences of the DNA in the chromatin, and to that attachment of the CENP-A. But when that normal chromosome, normal centromere is silenced, what will happen? The point to which centromere A, CENP-A is attached, centromeric protein A, not centromere A, centromeric protein A, CNP-A, or CEN-H3 is attached, that starts functioning as a centromere, irrespective of the sequence of nucleotides. That is, even if there is not repeated sequence there, that is, satellite sequences are lacking there, even then the attachment of the CNP-A will induce that segment to function as centromere. So what is important for functioning as a centromere? It is attachment of CNP-A or other variants like CID, CSC4, CNP1, whatever you may call. And if you want to refer all these collectively, it would be better to call them CNH3. So if CNH3 has got attached to a point other than the repeated sequences, which was in normal case functioning as the centromere, the previous point will be silenced and the newly attached point will begin to act as the centromere. So here the centromere marker is the CNP-A, and even if the normal chromatin arrangement that is found in the normal centromere is not present, the mere attachment of the CNP-A to any point, any new point of the chromosome will induce it to act as centromere. So that point, that new point, which has begun acting as a centromere, is called neocentromere, whereas the previous point, which no more acts as the centromere, has been called silent centromere. So, Priya, it has nothing to do with the dominant and recessive. Rather, it has to do with the attachment of the centromeric H3. And if centromeric H3 has got attached to a novel point, the previous point or the normal point, which was acting as the centromere hitherto, it now no more acts as the centromere, and it will be referred as silent centromere, where the new point, which begins as centromere, will act as neocentromere. So it will act in sequence independent matter, and recent findings have suggested that the centromere structure and dynamics changes to octamer. So the attachment of new type of histone H3 will change the entire dynamics of the histone core, that is the CEN-H3 histone will act differently from the canonical histone, that is the histone found in the other regions of the chromosome. So, alteration of nucleosomal histone stoichiometry, it will facilitate non-histone proteins, attachment of the non-histone proteins, as we have seen that centromere is defined as the length to which proteins are attached and to which kinetochore is attached. So the attachment of CENP-A facilitates attachment of other proteins, which will facilitate the functioning of the nucleosome centromere. CENP-A directs reversal of handedness. The wrapping characteristics, you know, nucleosome is DNA wrapped around the histone core. So wrapping characteristics also change. The number of bases wrapped, you know that in canonical chromosome, as I have taught you earlier, that in canonical chromosomes, 146 base pairs are wrapped around the histone core. But here, the number of bases wrapped has got lessened. Similarly, in canonical histone, there is left-handed wrapping. Here, it has been suggested that there is the wrapping is right-handed wrapping. So the attachment will change the characteristic of the octamer, that is the characteristic of entire histone molecule. And so to distinguish it from the CNP histone, we call it canonical histone and CNP histone. Now, what is the difference? What is the difference between canonical histone and CNP-A histone? You see here, octameric histone H3 nucleosome. So this is canonical. This is the usual histone nucleosome, and this is the CENP-A. That is, in this case, H3, canonical H3 is found. That is H3, which has been found in the normal nucleosomes. That is present here in this case, and here you have got that H3 replaced by the CNP-A. CNP, as I have told you, is the centromere marker. So it has marked centromere, it has changed, it has imparted the property of chromosome on that part of the, sorry, it has imparted the property of centromere on that part of the chromosome. And so how does these, what are the important differences between these two structures? So you see here, that this is the canonical histone. That is the histone found in regions other than centromere, the usual histones, you can say. So you see that histone H3, H4, H2A, and H2B. As I have told you earlier, that H3 and H4 for octamer, 2 molecules each, and dimers of H2A and H2B get attached to the scaffolding. Sorry, H3 and H4, 2 molecules each will form tetramer. So the H3, 2 molecules of H3 and 2 molecules of H4 will form a tetramer, and it will form the core, it will form the scaffolding around H2A H2B dimer. 2H2A, H2B dimer will get attached. So here you have got 4 molecules. Here are 2 plus 2, 4 molecules. And so this will form the histone core, that is the octamer of histones. You see here that this is histone 2A. So histone 2 molecules of histone 2A is attached and 2 molecules of histone 2B. So these are histone 2A plus histone 2B. Here histone 3 plus histone 4. So this is histone 3 and this is histone 3. One molecule of histone 3, another molecule of histone 3, and here it is histone 2. So here is the 1, 2, 1, 2, 3, 4, 1, 2, 3, 4. So octamer around which DNA is wrapped. So this was all about the canonical histone octamer, but here you have got CNPA nucleosome. That is, this H3 has been replaced by this H3. This H3 is what? CNP-A, that is centromeric H3. And so you see here that instead of these two, you have got 2 molecules of this. So rest of them is same. Here you have got CNH3, here you have got CNH3 or CNPA, whatever you may call. And here you have got 2 molecules of histone 4 attached to this, and here 2 molecules of histone 2A and 2 molecules of histone 2B. So in this octamer, you see that what is the basic difference? The basic difference is that this H3, which were found in octameric histone H3 or the canonical nucleosome, had been replaced by this H3, that is CNPA in place of histone H3. Now, what is the difference? What is the difference between these two? You see, the main difference is the C terminal, these two histones, that is histone 3, the canonical histone 3, and the CNPA, that is CENH3, centromeric histone 3, they show similarity or convergence at the C terminal. That is the C terminal, that is the terminal where this carboxylic is C terminal, where the free COH group is there. So at C terminal, they show similarity, but this similarity is apparent at N terminal. I am comparing the two N terminals, you see here. The N terminal of CENH3, that is the H3 which is found at centromeres, and here the N terminal of normal or the canonical H3. So it is the difference of N terminals that makes the two behave in different ways. Although there are some other subtle differences also, but the main difference lies at the N terminal of the two. These differences lead to the change in character. So, as I have told you, that about 146 base pairs are wrapped around this structure. This histone octamer is wrapping about how many bases? 146 bases in 6.4 5 turns. That is, the turn around the octamer is about 6.5 turns. And so in canonical histones, the DNA is wrapped around the octamer. About length is about 146. Here it had been written 147. So about 146 base pairs are wrapped in 1.65 turns. 1.65 turns, mind you, 1.65 turns. But here you see that around 120 base pairs are wrapped. So main difference is here 146 to 147 base pairs are wrapped. Here only about 120 base pairs are wrapped. CNP has a divergent amino tail with distinct modifications, as we have compared that this amino tail of these two, that is CEN-H3 and canonical H3, differs. CNP-A H4 tetramer is more compact. Another difference is that here the tetramer is made by 2 molecules of histone H3 and 2 molecules of histone H4. But here the tetramer is made by 2 molecules of CEN-H3 or CNP-A and 2 molecules of H4. So this tetramer consisting of 2 molecules of CNP-H3 and 2 molecules of 2 molecules of H4. 2 molecules of CEN-H3 or CNP-A and 2 molecules of H4, this tetramer is more compact than the tetramer formed by the canonical H3 and H4. So these are the differences. I have a question regarding from Saurabh regarding CNP-A versus canonical. So you see, Saurabh, that in canonical histones, you have the usual octamers consisting of 2 molecules of histone H3 and H4, which form tetramer at this scaffolding on which 2 molecules of H2A, H2B impinge upon them, that scaffolding as 2 dimers, and so octamer is formed. But here in case of octameric CNP, in case of CEN-H3 nucleosome, what happens? That histone H3 has been replaced by CNP-A or CEN-H3, and this CEN-H3 forms the tetramer along with the 2 molecules of H4. And this CEN-H3 H4 association is more compact than histone H3 and H4 association. So I think there was a question from Pia also regarding the diagrammatic representation of canonical, and so I have devoted quite some time on this diagram, and let us have a view of the text too. So CNP is a variant of histone, alpha carboxy terminal of histone 4 domain. So CNP-A is similar to H3. I'm saying it is variant of H3. How? So the similarity between two resides at carboxy terminal. The fold at carboxy terminal is almost same in both the histones. But what is the difference? Differ from conventional or canonical, whatever you may call, histone 3 at N terminal. So I have I want to show you on the same screen. So here you have got that slide, which was exhibiting the structure, and we can shift here that this C terminal shows homology, that is C terminal is almost similar in case of histone H3 as well as CNPA, but N terminal is different. The condensed nature of the array may reflect strong self-self interaction. What I told you earlier is that here I said you that the interaction between the CEN-H3 and H4 leads to the compaction of the centromeric nucleosome. So nucleosome becomes more compact, and this compaction has got impact on the structural integrity of the centromere too. The nucleosomes, which are compact and they are arranged closely, they provide strength to the structure of centromere, since the centromere is subjected to pull from both the sides during the separation of chromosome, during the movement of chromosomes towards the poles in anaphase. So it should be stronger enough, its integrity should be such that it could withstand the pulls from either side. So N-terminal is different, and that N-terminals recruits kinetochore proteins to centromere, and here CATD, centromere, CNPA centromere targeting domain. CNPA centromere targeting domain. So this domain, this structure, three-dimensional structure, facilitates the arrangement of attachment of the proteins to the centromere required for centromere targeting and function. CNPA targeting domain, CATD, is required for function. Substantial structural differences between CNPA and CH4. The differences that I have mentioned, and additional differences are the bulge loop. There is loop 1. There is a bulge loop in CEN-H3, that is CENP-A and its other variants, which facilitates closure interaction with the H4. So as I mentioned you earlier, that CEN-H3 plus H4 interaction is more profound, and it leads to compaction of the histone octamer at the centromere. This compaction is facilitated by the bulged loop that is called L1 loop that generates different shape, and since it is oppositely charged, it facilitates closure interaction with H3, and that leads to the generation of a rigid interface with H4. That is, the interface between CEN-H3 and H4 is rigidly attached. And then, as I have told you, that the size of the centromeric nucleosome is also changed. It is also smaller because these heterodimers are compact, that is CNPA, H4 is compact. So rotated CNP-CNPA interface that compacts the overall size of the CNPA here. So I said earlier that the interaction between this centromeric histone, that is CNP-A, and the H4 that is more compact. It leads to the compaction of the histone core, and this compaction is facilitated by this bulge loop, and this compaction will lead to the decrease in the size of the centromeric nucleosome. Since it is arranged more compactly, it will lead to the decrease in the size of the centromeric nucleosome. So I think I'm quite clear, and I have reached to you regarding all these points. And I think there is no gray area. This is the domain which is called CENP-A, centromere targeting domain. So this is centromere targeting domain. It facilitates attachment. Active domain, it facilitates attachment within the histone fold domain is known to be required for centromere targeting and function. It facilitates the attachment of the CNP-A to the centromeric DNA and incorporation of further proteins to it too. Now, impact of CNP-A nucleosome. Many of you, including Shweta, roll number 8, has raised the question about impact. So what are the impacts of the replacement of H3 by CEN-H3? That is, what are the impacts? What are the changes which are caused by the replacement of normal or canonical histone H3 by the centromeric histone H3 or CNP-A? So, as you have seen earlier, it leads to compaction. It will lead to closure arrangement of the nucleosomes and adjacent nucleosome per centromere intersect clusters of H3 containing nucleosomes. So, compaction and cluster formation, that is, these nucleosomes which have centromeres, these nucleosomes which have centromeric H3 attached to them will facilitate the compaction of the nucleosome as well as formation of the closure clusters, and this clustering of the nucleosome will strengthen the centromeric DNA portion. The condensed nature of the array may reflect a strong self-self interaction that culminate in coalescence of discontinuous CNP nucleosomes on each centromere. So what happens that this compaction and this clustering will lead to the closure arrangement. They will coalesce, that is, they will become closely arranged. They will come to lie closely. They will form clusters, and condensed nature of array may impart to help maintain a structural integrity of centromeric chromatin. So as I told you earlier, that this clustering, this compaction will impart strength to the portion of the centromere, and that will facilitate the pull of the spindle fibrils on either side, and the centromere will not give in to pull. That is, centromere will be substantially strengthened to withstand the pull pressure of the spindle fibrils from either side. Then there is entry and exit site is less constrained. Many of the students have got confused about this entry and exit site. So let us see. Here in this diagram, you see here the canonical nucleosomes, that is the usual nucleosomes which are arranged. You see that here is octamer of histone core, and the DNA is wrapped around it. So here, these are the nucleosomes. This is the linker DNA. Similarly, you see here octamer of histone core with DNA wrapped around it. Okay, but what is the difference? The difference is in H3. Here, the H3 is CEN-H3, that is H3 found at centromere, which is different from the canonical, that is the usual H3. So it is, in human case, it is called CNP-A, centromeric protein A. So the difference between these two is that H3 in canonical, whereas CNP-A in centromeric nucleosomes. These changes have led to impacts. What are the impacts? Number one is compaction. As I told you, you see these two histone cores, these octamers with DNA wrapped around it, that is these two nucleosomes, they have come to lie closer to each other. Here they are distant. So this compaction results from the replacement of canonical H3 by CNP-A. Another point is that entry and exit site is less constrained. You see here the entry site, DNA is entering, and here the exit. You see here the entry and exit site is tightly bound. So it is more constrained. It is more compacted. The histone core, the octamer, is tightly bound by the DNA wrapped around it, but here it has less constraint, that it is not that tightly coiled. In case of centromere, the histone core involves replacement of canonical H3 by CNP-A. This leads to the compaction of nucleosomes, and it has its impact also on the wrapping of the DNA around the nucleosome, because in canonical case, the wrapping is tight. It is tightly wrapped, whereas here the noose is not that tight. And so I said here that entry and exit site is less constrained, indicated by uncrossed internucleosomal DNA. Here internucleosomal DNA has crossed each other. But here in this case of centromeric nucleosomes, the internucleosomal DNA, that is the DNA portion between the two nucleosomes, or the linker DNA, whatever you may call. So internucleosomal DNA has not crisscrossed. That is, it has not been tightly bound. That is, it has not been more constrained. So as far as the compaction between the two nucleosomes is concerned, the compaction is more than the canonical nucleosome, but the entry of and exit point of the DNA, which wraps the centromeric nucleosome, has not been tightly noosed. It has not been wrapped that tightly, and this loose wrapping is represented by here that the DNA has not crossed each other. So here the entry and exit points have crossed each other. So I think it is more than clear that the DNA at the entry and exit site is less constrained. Less constrained in which case? In case of centromeric nucleosomes, indicated by uncrossed nucleosomal DNA for CNP-A containing arrays. The increased flexibility at DNA entry-exit site may accommodate internucleosomal DNA paths unique to the centromere and or nucleosomal binding proteins that require increased local access. As I have told you that the centromere is represented by the primary construction to which CNP-A is attached. So attachment of CNP to that primary construction facilitates arrangement of an array of proteins, and to that protein plus DNA bound structure, that is the centromere, the kinetochore gets attached. So this loose news or this loose less constrained entry and exit point will facilitate the entry of some proteins here, some proteins there. Had it been tightly noosed, as in this case, the entry of proteins will be hindered, will be inhibited, but here entry of proteins will be facilitated. It will allow easy access to the protein, and you know that attachment of those proteins are necessary for the centromere to act in that particular point, to act as a nodal point for the attachment of kinetochore and subsequently attachment of spindle fibers to it. So this point is clear that entry and exit site is less contained, and this loosened entry and exit site will increase access to the DNA, access of proteins to the DNA or to the histone. So the impact of CNA is impact of attachment of CNP each. The impact of attachment of CNP-A is what? What are the impacts? Number one, compaction of the nucleosomes. So it will lead to the clustering of nucleosomes too, and it will impart the clustered or the coalescence of nucleosomes will impart strength to the centromeric region, integrity to the centromeric region, so that it could withstand the pull from the either side. Number one. And number two, the loose noose around the histone cores will facilitate the entry of proteins which are necessary for imparting functionality to the centromere. Other variants of CNP-A nucleosomes. So the most common case of centromeric nucleosome, the histone H3 is replaced by centromeric H3, that is CNP-A or CENP-A or its variants. But there are other histone 3 variants too. So replacement of canonical H3 by CNP-A, which binds to histone H4 to form H3H4 dimer, the latter binding with H3H4 dimer to form tetramer. That is, two dimers bound to tetramer. Two dimers, each made up of H3, CEN H3 and H4. So one dimer is made up of one CEN H3, that is CENP-A, and another H4. So CENP-A plus H4, one dimer, CENP-A plus H4, another dimer, and these two dimers form tetramer, ultimately forming homotypic tetramer. That is, the two are identical. Two H3 are identical, they are CEN H3, and two H4s are identical, containing two copies of CENP-A is not the only variation. So the most profound variation was replacement of canonical H3 by CENP-A or CEN H3. But this is not the only variation. There are other variations too. This is the most common difference from the canonical nucleosome, but some other variants have also been reported. And in most recent work, 10% of human centromeric nucleosomes form a heterotypic octamer containing CNP-A containing histone and one canonical H3 containing histone. That is, here the octamer was being formed by CNP-A and H4, two dimers of CNP-A and H4. But in some cases, in 10% cases, that CNP-A histone, that is the centromeric histone, form dimer with canonical H3. In normal centromeric cases, in most of the centromeric cases, what happens? That centromeric H3 is canonical H3. In most of the centromeric cases, the canonical H3 had been replaced by the centromeric H3, that is the CENP-A. But in 10% of the cases, what happens that the centromeric H3, that is CENP-A, is also present along with the canonical H3, that is the normal H3 plus centromeric H3. Both are present, and centromeric H3, which was forming dimer with H4, forms dimer with H3, canonical H3. So here you can say that a dimer is being formed by centromeric H3 and canonical H3. This is an aberration. It has been found in 10% cases. In most of the cases, what happens? The centromeric H3 forms dimer with H4, and two such dimers come to lie together to form tetramer. But here, in 10% of the human centromeric nucleosomes, CENP-A containing histone forms dimer with canonical H3, and centromeric H3 plus canonical H3 form dimer, and two such dimers will form tetramer. Furthermore, work in melanogaster, Drosophila melanogaster, has found that different composition of centromeric nucleosomes are present. A nucleosome containing at least 2 copies of centromere identifier. This is CID. Some of you have raised question about what is CID. So CID is nothing but the variant of CENP-A. That is, CID is the CENH3 of Drosophila melanogaster, homologue of CENP-A. So in case of human, what is CENP-A? In case of CID, sorry, in case of Drosophila melanogaster, it is CID. So CENP-A protein, which imparts centromeric property on a length of this chromosome in human beings, is replaced by CID in Drosophila melanogaster. That is, it is the CID, centromeric identifier domain. It is the CID which imparts centromeric property on the chromosome of the Drosophila melanogaster. So a nucleosome here, one variation is that in 10% of human beings, both CENH3 is present along with the canonical H3. That is, CENH3 forms dimer with canonical H3. In normal cases, in most of the centromeric nucleosomes, what was happening? CENH3 was forming dimer with H4, and those dimers come together to form tetramers. But in some cases, in 10% of the human cases, CENH3 is forming dimer with canonical H3. In Drosophila melanogaster, two copies of CID are present. There is a hemisome structure. One copy each of H2A, H2B, H34, and CID may be present. So 2 copies form octamer nucleosome, but 1 copy each, that is tetramer, will form hemisome. And hemisome, it has been speculated that might be a stage which precedes the octamer formation. So it might be a stage, transitory stage in the formation of the octamer. It may not be an independent stage. Nevertheless, it had been found. Then there is another variant of H3 called H2AZ variant at the centromere. CENP-A containing nucleosomes contain canonical H2A, whereas the variant H2AZ is associated with nucleosomes containing H3K4ME2 and in the centromere of human and mouse. So this nucleosomes, they are methylated at point 2. We shall be discussing this H3K4ME2 and H3K9ME3 in subsequent slides. while we will deal with the epigenetics of the centromere. So in a passing way, I'm mentioning here that these are the methylated structures, nucleosomes, which have undergone methylation, and these nucleosomes have H2AZ, that is another variant of canonical H2, that is H2AZ. H2AZ containing nucleosomes are more resistant to condensation and form a boundary between heterochromatin and euchromatin. It has been seen that such nucleosomes are found at the boundary of H2A, at the euchromatin and the heterochromatin. So what are the other variants? The first variant was that H3, CENH3. The first variant was that CENH3, centromeric H3, that is CENP-A, was forming dimer with canonical H3. In normal case of centromeric nucleosomes, what happens? That CENH3 forms dimer with H4, and CENH3, H4 dimers, two dimers come to join together to form octamer. So this is homotypic octamer, that is, it have H3 plus H4. But in some cases, 10% of the cases in human beings, the CENH3 forms dimer with H3, canonical H3, rather than the H4, as was case in normal centromeres. Then another difference is in case of Drosophila melanogaster, the CID, that is the homologue of CENP-A, may be 2 copies of CIDs instead of 1. One nucleosome contains 2 copies of the CID. Here it was CNP-A. CID is the homologue of CNP-A. So in Drosophila melanogaster, 2 copies of CID is present. There are differences. Number 1, 2 copies may be present, as happens in case of normal centromeric nucleosome, that the 2 copies of CENP-A are present and 2 copies of CENH3, canonical H3, H4 is present. But in case of Drosophila, hemisomes may be formed also, and it will have 1 copy each of H2A, H2B, H4, and CID. In normal case, what was the composition? 2 copies of CIDs, 2 copies of H4, 2 copies of H2A and H2B, 2 copies each. But here, and the variation is, there is only 1 copy of each, that is, instead of octamer, it is forming tetramer, and such tetramer is called hemisome, and hemisome might be a stage in the formation of octamer. Now, there is another variation too, and that is in certain methylated nucleosomes, normal H2A, that is canonical H2A, has been replaced by another variant, which is called H2AZ, and it has been found that H2AZ containing nucleosomes are found at the boundary of euchromatin and the heterochromatin. So here you see hemisome and heterotypic nucleosome. Hemisomes 1, 2, 3, 4. 1, 2, 3, 4. So instead of octamer, it is a tetramer with DNA wrapped around it. 1 copy of CNP-A, H4, H2A, and H2B. So instead of an octamer, it is a tetramer, and it has been referred to as a hemisome. But here, the main difference is that in the homotypic CNP-A H4 nucleosome, that is the nucleosome which is found at the centromeres. Most of the nucleosomes, they have CNP-A and H4. 2 molecules of CNPA and 2 molecules of H4 forming the octamer. But in this heterotypic nucleosome, CNP-A form dimer with canonical H3, that is H3 which was found at loci other than centromeres, that is the canonical H3, which was the normal H3, usual H3 in the nucleosome, here forms bonding with CNP-A. CNP-A was variant of H3. It has replaced H3 in most of the chromosomes, but here in some cases, heterotypic cases, CNP-A as well as H3 both are present and they form dimer and subsequently octamer. So 1 copy of CNP-A, 1 copy of H3, 2 copy of H4, H2A, H2B may represent a subpopulation of centromeric nucleosomes. That is, it is not the main population, rather a subpopulation could occur at specific stages in the cell cycle during CNP assembly and after DNA replication. It has been presumed that it is present during the specific stages of the cell cycle, maybe during CNP-A assembly or at the time of DNA replication. So today we have dealt with the significance of centromere, and while dealing with the significance of centromere, we have delved deeply into the structure of the centromere. And the centromere, if I may sum up, is nothing but it is a primary constriction which holds the two sister chromatids together, and to it certain proteins are bound, and to those protein bound centromeres the kinetochores are bound, and to those kinetochores the spindle fibres are attached which pull the DNA up, pull the sister chromatids apart during this cell division. So the centromeric property is imparted by attachment of one protein, that is the CENP-A, centromeric protein A, which is the variant of the canonical H3, that is H3 which was found in usual nucleosomes elsewhere, that is other than centromere, had been replaced. That H3 had been replaced by CENP-A, and this attachment of the CENP-A chromatin imparts centromeric property. So usually the chromatin, we have not studied the structure of chromatin at the centromere. We shall study in the next portion. So the attachment of the centromeric protein A to the region of the DNA imparts centromeric property, and so the CENP-A, you may call it the main centromeric marker, that is wherever it attaches, even if the sequence of nucleotides differ, that is the randomly repeated sequences are not present, even at that point of attachment will lead to that point will function as, henceforth function after the attachment of the CNP-A will function as the centromere, and that new functioning centromere is called neocentromere, but the previously present centromere which no longer functions is called silent centromere. So it was the incorporation or attachment of CENP-A which imparts the centromeric property to a particular stretch of DNA, and this attachment of the CENP-A containing nucleosome will facilitate attachment of further proteins, which will further facilitate the attachment of kinetochore, and this attachment of kinetochore will lead to the attachment of spindle fibrils. So this centromere attachment of CENP-A imparts many changes in the nucleosome. It leads to the compaction of the nucleosome. It leads to the loosening of the noose of the nucleosome DNA around the nucleosome, and this loosened DNA entry and exit point, that is not constrained DNA exit and entry point, it will facilitate the entry of proteins. And you know that the centromere formation will only be successful only when the proteins are attached. That is, CENP-A containing nucleosome facilitates attachment of more proteins called CCPA. The CCPA proteins are attached, a group of proteins are attached, and those proteins imparts not only the biochemical property for attachment of kinetochore, but also imparts physical integrity to the centromere, so that centromere could withstand the pulling apart of the two. Can withstand the pulling pressure of the two poles of the centrosomes, the spindle fibers, which are pulling apart the centromere. So in this part, we have dealt with the characteristic of centromere, how would we define centromere, and what impact the replacement of canonical H3 by CNP-A has been on the centromeric region, and it leads to the compaction, it leads to the tightening of noose. And we have also studied other variants of the CNP-A, that is centromeric nucleosomes, in which CNP-A forms dimer with the canonical H3. In most of the cases of this centromere, CENP-A form dimers with H4, but in some cases CENP-A forms dimer with normal H3. And in there was another variant in which instead of octamer, a tetramer is formed around which DNA is wrapped, and such structure is called hemisome. And in another dimer, H2A variant is present. So these are the variations, but the mainstay is that the centromeric attachment of the centromeric protein A to a particular region of the DNA imparts to a particular region of the chromosomes, imparts the centromeric property to that chromosome, and it facilitates attachment of more proteins. And those proteins strengthen, not only imparts functional specificity to the centromere, but also strengthens the centromeric region so that it could withstand the pulls from the both sides, and it carries the entire chromatid with it, and the fidelity of the segregation is maintained. So that was all about in today's lecture. And in the part 2, we shall be dealing with the different types of centromeres. And in that lecture, we will encounter holocentric chromosomes. And in some cases of the holocentric chromosomes, that is the holocentric chromosomes found in insects, they lack CENP-A. So here, there is an important exception. Up till now, I have been stressing and overstressing the attachment of CENP-A to a particular region of the DNA or the chromatin, which will impart centromeric property to that particular region of the chromosome. But there are certain cases too. About 16% of the eukaryotic biodiversity represented by the holocentric insects, and in holocentric insect, CENP-A is absent altogether. And you have raised these questions that how the holocentric chromosome has evolved and how CENP-A is absent, and how in the absence of CENP-A, the centromere is functioning, that is, the kinetochore is attached to the centromere, and how that centromere facilitates the segregation of proteins. So we shall see all these in the next lecture, which will be following in a day or two. And so I must give I expect a feedback from all of you about this lecture, and you must raise the points which have inadvertently not been clarified in this lecture, or the gray areas, if left in this lecture, must be raised so that I could recount those points in the next lecture, or clarify those points in the next lecture. So it was very vibrant class. I enjoyed being with you, although in the offline I'm unable to see you, but I visualize that my entire class is present before me, and I think you nodding to my points and your facial expressions I could also visualize. And so I tried my level best to make this offline class make this online class into an offline class. Thank you. Thank you very much.